Carbohydrates are essential for aerobic respiration. In fact, your brain can only run on glucose. So even if you don't ingest carbohydrates, you have to synthesize them in order to survive. Let's get into it.
So some key terms just to start off today's lesson. First of all, in A-Level Biology, you need to know about monomers. Now, monomers are single molecular units, such as glucose in starch. A polymer is a long chain of monomers that have been chemically bonded together. And key examples of that include starch, cellulose, and glycogen. Polymerization is the formation of a polymer which involves chemical reactions known as condensation reactions. Monosaccharides are single sugar units. Clear examples of those include glucose, fructose, and galactose. Disaccharides are where two monosaccharides have been joined with a glycosidic bond. Polysaccharides are when more than two monosaccharides have been joined by a glycosidic bond. Next up, condensation is a reaction that forms water and bonds. So it's a reaction that forms bonds releasing water. Hydrolysis is a reaction that breaks bonds using water. So think of hydro-water lysis splitting, water splitting using water to break a bond. Triose is a free carbon sugar. Think about a tricycle having free wheels. Pentose is a 5-carbon sugar. Think about a pentagon having 5 sides. Hexose is a 6-carbon sugar. Hexagonal 6 sides. A glycosidic bond is a bond between sugar units, and in this case they'll have an oxygen shared between them, and I'll show you a more in-depth look of how that can come up in the AQA A-Level Biology exam soon. Lactose is a disaccharide made up of glucose and galactose. Sucrose is a disaccharide, di meaning two. Think about carbon dioxide that has 2 oxygens. Disaccharide, 2 sugars, and it's made up of fructose and glucose. Maltose is a disaccharide made up of 2 lots of alpha glucose joined with that glycosidic bond. The benedicts test is a test for reducing sugars, and what happens is it goes from blue to brick red if there's a positive result, and that brick red will be a precipitate, which is when a solid forms from a liquid. Green would indicate low concentrations of reducing sugars. A reducing sugar is basically all monosaccharides, and there are certain disaccharides, for example lactose and maltose, that can also act as reducing sugars too. A non-reducing sugar includes sucrose. That's the main example you need to know for AQA A-Level Biology. Iodine, or potassium iodide, is a solution that's brown, but when added to starch, it will go from brown to blue-black. And finally, cellulose, starch, and glycogen are 3 key polysaccharides that you need to know made up of glucose. Monomers and polymers. So on the screen, I've got a polymer here, which I'm just representing it as starch. And if we look at it, a polymer is made up of many repeating molecules. A monomer is just one individual molecular unit. So a classic example of a polymer is starch, glycogen, or cellulose that you need to know about for AQA A-Level Biology. But an example of a monomer is something like alpha glucose, and alpha glucose is the monomer that makes up starch. Now you can see these circles, they're going to represent single units such as glucose, and the lines between them are representing covalent bonds, and in the case of carbohydrates, the bond between the monomers is a glycosidic bond. Now monomers are single repeating molecular units. Like we've said, things like monosaccharides, which is, think about a monobrow, single eyebrow. Monosaccharide is a single sugar unit. But it could also be something like a nucleotide or an amino acid. Watch my video on protein structure for more information about amino acids. Polymers are long chains of monomers joined together by chemical bonds. And polymers are formed via, like we said in the key terms section, polymerization reactions, and starch, glycogen, DNA, cellulose, polypeptides, they're all classic examples of polymers. Now, two of the key monomers you need to know about, the monosaccharides, because this lesson's all about A-level biology carbohydrates, the monosaccharides you need to know about are alpha and beta glucose. Now, they're very similar. So we have, 6 carbons, carbon 1, 2, 3, 4, 5, and then just look above the ring structure for carbon 6. And they've both got an identical kind of ring structure, but there is one key difference, and that difference comes here. So when we're looking here, I will just grab my pen. Around carbon 1, so here, the hydrogen and the hydroxyl are reversed, whereas on beta glucose, you can see they're the other way around. So in alpha glucose, the hydrogen is at the top of carbon 1, and on beta glucose, we have a hydroxyl group at the top of carbon 1. Now think of hydroxyl, hydrogen, oxyl, oxygen, because it's made up of hydrogen and oxygen, okay? Now that's the only difference between these two molecules, but it does lead to some really quite striking structural differences when we look at cellulose and starch, and I'll give you more information about that now. So, both have the formula C6H12O6, and as we've said, the hydrogen and the hydroxyl group is reversed at carbon 1, and that's the difference between the two. Now, alpha glucose makes up maltose, glycogen, and starch, whereas beta glucose makes up cellulose. Now, I've drawn a diagram here representing a condensation reaction between two molecules of alpha glucose. And we know it's alpha glucose because, again, just to recap that, if I grab my pen, we can see here the hydrogen is at the top of carbon 1. And if we look at this glucose here, the hydrogen is again at the top of carbon 1. And we could number all the carbons, so we've got carbon 2 there, carbon 3 there, carbon 4 there, carbon 5, and carbon 6. Now, you need to know this inside and out for A-level biology. It's clear in the specification that AQA want you to understand the structure of both alpha and beta glucose, so that's why I've emphasized it here. Now, if we look at this diagram, at this image here, we can see that there's the OH at the bottom of carbon 1 here, and there's also an OH, a hydroxyl group, at the bottom of carbon 4. Now, a condensation reaction releases water, so what we do is we remove water, and remember water is H2O, so 2 hydrogens and an oxygen, get removed, and that goes over here. And you can see on the left in yellow, I've got my H2O there. And because that water's been removed, it leaves us this oxygen, which I've just represented in pink there. Now, after this bond has been formed, we're left with a shared oxygen. And that shared oxygen there is what's called a glycosidic bond. Now, the molecule that's formed as a result of this is a disaccharide, and that specific disaccharide is maltose. Maltose is broken down by the enzyme maltase. More on that in a video I'm going to release on digestion. So, we have alpha glucose plus alpha glucose going to maltose plus water. So just to summarize this, condensation forms bonds, releasing water, whereas hydrolysis is the opposite of this. So we could show hydrolysis here, where we'd have to actually add water, and it'd be the other direction. Now, hydro meaning water, lysis meaning splitting, so splitting using water. So how do we test for reducing sugars then? Well, what we have is something called the benedicts test. Now, the benedicts test is where we add a sample to a test tube. If it's a solid food, then we might need to crush and grind it and add some distilled water to put it in solution, or at least in suspension. The next step would be to add benedicts reagent to the sample and heat it to around 80 to 90 degrees Celsius. The AQA A-Level Biology mark scheme will be looking for you to say heat it. If we get a brick red precipitate, remember a precipitate forms where a solid forms from a liquid, that will mean we've got a high concentration of reducing sugar. Green, yellow or orange will be a lower concentration of reducing sugar, still a positive result. Now this is an example of an indication of the Benedict's test, and we can see if there's 0% sugar on the left, it will be blue because that's the colour of the reagent. But the stronger the colour, the more it gets to brick red, the more precipitate is formed and the more reducing sugar is present. Now, you also need to know the Benedict's test for non-reducing sugars, so that can come up in the exam. And in order to do that, the first thing you will do is do the Benedict's test for reducing sugars to rule out there being any reducing sugars present. Once you've established that there's no reducing sugars present, so if negative, you will then add a dilute acid to the sample, which should hydrolyze bonds. Remember, add water to break bonds. Then what we'll do, once we've heated it with the acid, is neutralizing it using an alkali such as sodium hydrogen carbonate. And AQA give the example of sodium hydrogen carbonate in some of their past paper questions, so that's why I've included it here. You will then add Benedict's solution to the sample and heat it. And this time, if you get a brick red precipitate, that will show that you're positive for non-reducing sugars, because there were no reducing sugars in the first test, but it was positive in the second test, proving the presence of non-reducing sugars. So here again, we can see our colour chart showing the negative to positive results. Glycogen is a large polysaccharide that you need to know. Now, it's used as an energy store in animals, and it's stored in the liver and muscles. Now, glycogen is made up of alpha-glucose monomers, and it's highly branched. And my students sometimes find it quite tricky to know about the branching, so I've included it nice and clearly for you here. A 1-to-4 linkage will be found here in the straight portion of the chain. So if I just grab my pen, let's number our carbons again, because remember, this is key. I always advise my students to learn how to do this. So we've got carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. So we can see we've got our 4 there. And then on this side, 1, and you always go clockwise, 2, 3, 4, 5, 6. So that's a carbon 1 to carbon 4 linkage. Now, where glycogen branches, we have carbon 1 to 6 linkages. And we can see that here. So let's go round. You go clockwise from the oxygen. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. And this side, clockwise from the oxygen, carbon 1. So we have carbon 1 to 6 linkages where glycogen branches. Now, glycogen is incredibly branched. Starch is the polysaccharide used as an energy store in plants. So animals, glycogen, plants, starch. Now, it's made up of alpha-glucose. So remember, the hydrogen is at the top of carbon 1. Amylose and amylopectin are long chains of alpha-glucose found in starch. Now, amylose is unbranched, and we can see that in the diagram on the bottom right here. It's long, and it only has carbon 1 to 4 linkages. So there's no branches. But what it is coiled, which is going to mean it's compact, and we can store more glucose in a plant cell that way. Amylopectin, on the other hand, is branched, and that gives more ends for enzymes to hydrolyze, making glucose release faster. And we can see in amylopectin, it's got carbon 1 to 4 and carbon 1 to 6 linkages. So that's where wherever it branches, we've got a 1 to 6 linkage, just like I showed you in glycogen. Now, starch will not affect the water potential of cells because it is insoluble, meaning it won't dissolve in water. And starch is stored in starch grains. And we can see here some starch grains in potato cells. Cellulose is the next polysaccharide you need to know about for AQA A-Level Biology. It's made up of beta-glucose, and remember, at beta-glucose, carbon 1 has the OH at the top. Now, it contains long, unbranched chains linked by hydrogen bonds. Now, these are weak individually, but they're very strong in numbers, And DNA also includes hydrogen bonds, so you'll learn more on that in that topic. A number of cellulose chains together will form a microfibril. And cellulose is found in plant cell walls, given a structural and supportive role. So that's everything for today, guys. Don't forget to like, comment, and subscribe. I want to hear from you. What do you want to hear next? Drop it in the comments, and I will see you in the next one.